Hi everyone, Dr. Hinkey here, and today we will be covering Chapter 8 on cellular respiration. So yesterday we covered, or in the previous chapter, Chapter 7, we talked about photosynthesis. You're going to see a lot of similarities here in the processes, which is sort of reversed. So our outline for the chapter, we'll look just a broad overview of cellular respiration. And then we'll look at the steps of cellular respiration, glycolysis, which takes place in the cytoplasm. And then we'll look inside the mitochondria at the other steps, at the preparatory reaction, the Krebs cycle, and the electron transport chain. Then we're going to look outside at fermentation. We're going to reverse the order of these because I want to get the complete process of cell respiration done first, before we step back to look at the anaerobic process of fermentation. And then we'll finish with an overall look at metabolism. So cell respiration is a process that breaks down nutrients and uses the energy from breaking down organic molecules, primarily glucose, to produce ATP. That's the energy molecule the cell is able to use. We're going to use oxygen, so that's an input, and we are going to produce the waste product of carbon dioxide. So you remember those two are reversed in photosynthesis, where carbon dioxide is the input and oxygen is the waste product. So cellular respiration is an aerobic process. That means it requires oxygen. And usually when we're talking about cellular respiration, we're looking at the complete breakdown of glucose to carbon dioxide and water. So we're going to take glucose, an organic molecule, C6H12O6, with lots of carbon-hydrogen bonds. We're going to end up with two inorganic molecules. No carbons will be left attached to any hydrogens, no hydrogens left attached to carbons. We're going to extract all of the energy from the glucose molecule. So when we do this, we're going to do this in a stepwise fashion. So if we go back to our laws of thermodynamics, the first law states that energy can't be created or destroyed. It can only change forms. So we are going to change one form of chemical energy, that potential energy stored in the glucose and the carbon-hydrogen bonds of glucose, and we are going to convert that to usable forms of energy, to the energy contained in that third phosphate bond, this bond between the second and third phosphate of ATP. That's the energy our cell is able to use. And we're going to do this stepwise. We are not just going to do one step to separate all the carbons from all the hydrogens in that glucose and release all the energy at once, because if we did that, we know our second law of thermodynamics tells us every time we convert from one form of energy to another, we lose some energy as heat. So that energy is now heat energy, which dissipates too quickly for us to use it for work. So if we broke every one of those carbon-hydrogen bonds in one step, we would release the equivalent energy of like a little baby nuclear bomb every time we broke down glucose. So instead, what we're going to do is break down step by step by step by step in little transactions. So we're going to have a metabolic pathway, even though our equation shows one arrow. We remember when we learned about enzymes, the metabolic pathways, we do small conversions, A to B, B to C, C to D, D to E, and each one of those has its own enzyme related to it. So we're going to do this step by step. So rather than one big breakdown of glucose, we're going to do many tiny ones, and that allows us to release a lot less heat, so it's much more energy efficient. To do this, we are going to use some enzymes. There can be lots of enzymes involved at each step, but some that are involved at almost every step are our oxidation, reduction, enzymes and coenzymes, NAD and FAD. If you remember in photosynthesis, we used NADP. So there's a big long name for NAD and FAD and NADP. If you remember these initials, the acronyms for these, you'll be good because you probably won't remember the full name. And the way I remember which one is the one with the P in it goes with photosynthesis, NADP. The other two, NAD and FAD, are going to be involved in cellular respiration. So here's our equation for cellular respiration. Glucose and oxygen are inputs. Carbon dioxide and water are our outputs from the process, so our reactants, our products, and we get energy in the form of ATP out. Electrons are removed from the substrates. Remember, that's a synonym for reactants when we have enzymes involved in a chemical reaction. Those electrons are removed from the substrates and received from the substrate of glucose, received by oxygen, which is going to combine with hydrogen to become water. Okay, sure. What does that mean? Remember when we talked about photosynthesis, whenever we're using the word electrons in these descriptions, in these processes, those electrons are the ones that we find in hydrogen. Remember, our hydrogen with an atomic number of 1 and atomic mass of 1 has 1 proton, no neutrons, and 1 electron. When we separate that electron from the proton, that's the one that's containing all the energy we're going to capture and use and move around to do these conversions. So if we remove an electron, lose an electron is oxidation, gain an electron is reduction. So we look for the hydrogens, follow the hydrogens. Lose an electron. Well, you have to have one to lose to begin with. No hydrogens here, so glucose must be the molecule that's going to be oxidized. And if I take away those hydrogens, I'm left with carbon and oxygen. So glucose will be oxidized and become carbon dioxide. My oxygen has no electrons, no hydrogens, so it can gain them. Gain of electrons, reduction. If I gain an electron, a hydrogen here, I get H's and O's. I'm going to reduce my oxygen into water in this process. So this is pretty much the exact reverse. If I flip this arrow around the other way, and these become my reactants, or my inputs, carbon dioxide plus water, and oxygen and glucose, then I have photosynthesis, and I have the reverse. In photosynthesis, water is oxidized into oxygen, carbon dioxide is reduced to glucose. So these are reverse processes. So look at cell respiration. Cell respiration is going to occur in the cell. Oxygen and glucose are going to enter. Hydrogen, water, and carbon dioxide are going to be released. It happens in the mitochondria, and both plants and animals have mitochondria. Remember, only plants have chloroplasts, but all living organisms need to have a way to release energy, to convert energy to a usable form, to ATP. And so in eukaryotes, we have this organelle, a membrane-bound organelle, the mitochondria. Prokaryotes that don't have membrane-bound organelles, that don't have this, they actually just have on their membranes, remember this internal membrane, this was originally a prokaryote that was engulfed by a larger prokaryote. And so along this membrane, this inner membrane was actually the plasma membrane or the cell membrane of that precursor living prokaryote. And the membrane has proteins embedded in it. So for prokaryotes, these reactions just take place on the plasma membrane. In eukaryotes, we're going to do all those on this inner membrane of my mitochondria. So just a little bit to set the stage here of what we need for cellular respiration to take place, we need our coenzymes that are going to be our hydrogen taxicabs again. So same function as NADP had in photosynthesis. We now have NAD, nicotinamide adenine dinucleotide. Adenine. Oh, we've seen adenine. That's our nitrogenous base. It's a dinucleotide, so 2 nucleotides. And nicotinamide tells us sort of a little bit about its chemical composition. So this is a coenzyme of oxidation reduction. It's oxidized when it loses electrons. So if it's carrying an electron, remember electron means hydrogen. If it's carrying one, it will lose it and become NAD. Gets oxidized. And when it picks up that hydrogen, it's reduced. It accepts the electron that goes with the H. And we're just going to keep recycling those, just as we did with NADP in photosynthesis. We're going to do with NAD, NADH. Pick up the hydrogen passenger, drop it off. Go back to pick up another hydrogen, drop it off. So we're going to recycle these. It's going to make this a very energy efficient process, or a conservation of mass. We're going to be very efficient about recycling our particles so we don't have to build new ones. And then FAD is flavin adenine dinucleotide, also a coenzyme, does the same thing. It's another taxicab. So our phases of cellular respiration, we actually have 4 steps in cellular respiration. The first one is glycolysis, where we break down glucose into 2 molecules of pyruvate. So glucose, C6H12O6, has 6 carbons. Pyruvate has 3 carbons. So we're just going to split our glucose into 2, and we're going to take away 2 of those hydrogens. In this process, we form some ATP. So we get a little bit of energy from this. We don't use any oxygen for this process. So this is an anaerobic process that gets us started. We'll use our oxygen later. The second step is the preparatory reaction. Some textbooks show this as a separate step. Some just sort of incorporate this, wrap this right up into the third step, the Calvin cycle. The Krebs cycle. Calvin cycle is photosynthesis. Don't get our cycles mixed up. So the Krebs cycle, or the citric acid cycle, is part of cell respiration. But the prep reaction is going to convert our pyruvates to a form that allows them to enter the mitochondria. So they become oxidized. So they lose an electron so that they can go into the mitochondria. They're going to lose that electron, the hydrogen, by giving it up to the hydrogen taxicab, the electron taxicab, NAD, and forming NADH. And in this process, we'll release some carbon dioxide. A little bit of a waste product will be released. The third step is the citric acid cycle, or the Krebs cycle. Now we are inside, so the prep reaction let us enter the mitochondria. So we're in the matrix of the mitochondria. That's the inner equivalent of cytoplasm, the aqueous solution on the inside of the mitochondria. And we are going to use our coenzymes, our carrier molecules, NAD and FAD, to pick up some hydrogens. This is a cycle. Remember the Calvin cycle, I compared it to a clock. We start at midnight, we change to 1, we change to 2, we change to 3. So we have all these intermediate hours, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. At 11, our final intermediate hour, we don't keep going, it's a circle, we come back to midnight. So that final change brings us right back to where we started, and then we go around again. So we have a series of reactions that bring us back to where we started each time. A new input, that final hour, brings us back to our starting point. Here, our final step brings us back to where we were, and we put in one more molecule of pyruvate to start the cycle again. We have 2 turns of this cycle for each glucose molecule that we started with. If you think about that, when we enter this cycle, we no longer have a glucose molecule. We've split that into 2 pyruvates. So we go around once for each pyruvate, and we get 2 ATP molecules per glucose molecule, or 1 per pyruvate that came into this. So we're getting small bits of energy, ATP, usable energy, from these steps. We don't get any ATP from the prep reaction. We're just preparing our pyruvate there. But so far we've generated 4 ATPs. Not a whole lot, but we have some energy. And then the final step, this is our big payoff in terms of ATP, the electron transport chain. Remember, the electron transport chain in photosynthesis was where we generated the ATP, the energy that was going to let us put our 2 small inorganic molecules, carbon dioxide and water, put them together into the large molecule of glucose. So here, again, our electron transport chain is going to produce ATP. Just like the electron transport chain of photosynthesis, this is on the inner membrane of our organelle. Now, not the chloroplast, but the mitochondria. And that inner membrane is called the cristae of the mitochondria. It's the inner membrane. And I have a series of carriers of those proteins along that membrane that are going to bounce those electrons along. And I am going to use those hydrogens that were carried over in the taxis, in my NAD and FAD taxis. Their job is to carry those hydrogens to the electron transport chain, drop them off, where the electron transport chain will bounce them through the series of proteins and molecules on the membrane that will let us use that, the energy contained in those electrons. As it passes the electrons from one carrier to the next, we have a series of oxidation reduction reactions. That electron comes in with a very high energy state. There's a lot of tension, a lot of energy in it wanting to drive back to its hydrogen proton. And each bounce releases a little bit of that energy until it's back to its low energy state. We let it return to its hydrogen. We capture that energy to maintain a concentration gradient by keeping it separate from the hydrogens, just like we did in our photosynthesis. And at the end of the process, the electron transport chain alone is going to produce 32 to 34 molecules of ATP through chemiosmosis. We looked at that in photosynthesis, where we maintain that concentration gradient of hydrogens on one side until we finally let them pass through ATP synthase to rejoin their electrons and to join up with oxygen again. So here is my chemiosmosis. We're kind of doing the reverse of what we did in cell respiration. There we started with the electron transport chain. Here we're going to finish with it. So step by step, our first step takes place outside of the mitochondria. So it's out here in the cytoplasm. And I start with the glucose that I'm going to convert into 2 pyruvates. I'm going to have some NADs that are going to pick up some of the hydrogens from my glucose and carry them to the electron transport chain. I have to kickstart this process by putting in 2 ATPs. I'm also going to start with 4 ADPs and 4 Ps, that this process will put together my ADPs and Ps to produce a total of 4 ATPs. Every time I do this, I need to put 2 ATPs in. So of these 4, 2 go right back into the process. This means I have a net gain of 2 ATPs for the cells to use. So look at glycolysis. It occurs in the cytoplasm. I'm not in the mitochondria yet. I'm not using any oxygen. There are 10 separate reactions, each one having its own enzyme. And the first thing I have to do is kickstart the process with those 2 ATPs. So here, throughout, everywhere you see something in blue, it's an input into a step. The greens are what comes out of each step. I put 2 ATPs in to start the process, and I put my glucose in. It's going to split. Oh, there's my G3P molecule, my ending molecule from photosynthesis. It's going to split into that. And then I'm going to oxidize G3P. I'm going to remove those hydrogens by having them picked up by NAD, which will become NADH. I end up with 4 ATPs at the end of this through substrate level phosphorylation. Right? So remember, phosphorylation, I haven't really talked about this, but this is how ATP works. It transfers those phosphates back and forth. It picks up phosphates from ADP plus P. It picks up phosphates to become ATP. It transfers those third phosphates in order to transfer its energy during chemical reactions. So an enzyme is going to put that ADP and a P together to make ATP. That's my substrate level phosphorylation. ADP is the substrate in this case, and it's going to have a P added to it. So I get 4 ATPs out, which really is a net gain of 2 because two of those are going to come back here and start the process again. And those G3Ps, when they lose those hydrogens to NAD, become pyruvate. This image is in your book. Don't let it break your brain. It's just showing each one of the steps, the phosphorylation. I break off an ATP. Those 2 ATPs get put in, and they phosphorylate my initial glucose molecule, which is then split into G3Ps, which then give up hydrogens, go through a series of steps. I am now going to use this phosphorylation to produce some ATPs. So I get 4 out after I put those 2 in. And I end up with 2 pyruvate molecules. What you really want to pay attention to in the textbook in these images is this. This is my accounting. What goes in each step, what comes out of each step. So glycolysis takes place in the cytoplasm. The inputs are 1 glucose molecule. It's a 6-carbon molecule, C6H12O6. I have 2 taxicabs that go in. And I have to kickstart the process with 2 ATPs. And then I have my broken ATP, which is broken into ADP and P, because this has gone and done work somewhere else, and now it's just waiting to be put back together so that it can go on to do work. My outputs are these 2 pyruvate molecules. So they're 2 3-carbon molecules. So I still have all my carbons here. 2 NADHs, because my taxi cab picked up their hydrogen passengers. When I start the process with these 2 ATPs, I break them down to ADP. Remember, those Ps got stuck onto the glucose molecule. So it's part of the pyruvate now. And that energy let me put these 4 ADPs back together with their Ps. So I got 4 ATPs out of the process. 2 of them keep coming back here. So I have a net gain of 2 ATPs. What happens to those outputs from glycolysis? Well, these pyruvates go to the next step, the preparatory reaction. My taxicab carries the hydrogens to the electron transport chain. This is what the taxis do, go back and forth, drop off their hydrogen passengers at the electron transport chain, and then come back to pick up more hydrogens. And my net gain of 2 ATPs, they're available. The cell can now use those. It has energy to do work. All right, so we'll move on to the prep reaction. Now, once that my NADs have been reduced and have the hydrogens, my pyruvates have been oxidized. They lost 2 hydrogens. They are now able, if oxygen is present in the cell, I'm not using oxygen yet, So those 6 oxygens that went in are not coming into play yet. I'm not using them. But as long as there's oxygen present, then those pyruvates are able to enter into the mitochondria. So I must have oxygen present to go to this next step, but I'm not going to use it yet. It just has to be present. So we consider this an aerobic step, even though we're not using the oxygen. It just has to be there, or else we can't enter the mitochondria. So my prep reaction is going to take place inside of the mitochondria. And inside the inner membrane, this inner membrane is called the cristae, and this aqueous solution in here is called the mitochondrial matrix. So we're going to take place in the matrix. Here's a close-up. So I have my double membrane. So this would have been, in the original, that endosymbiont theory, this would have been the membrane that surrounded my prokaryote that was engulfed from endocytosis. And this would have been the plasma membrane of my prokaryote. So I have a double membrane on my mitochondria. That inner membrane is called the cristae, and the matrix is the aqueous solution that's carrying all of the resources I need. All of those enzymes, coenzymes, all the factors that I need are dissolved in this inner matrix. So we're inside the mitochondria for the prep reaction. The prep reaction is sort of just the intermediate action between glycolysis and the citric acid cycle. So the end product of glycolysis, those pyruvates, enter into the matrix. The pyruvate is converted to a 2-carbon acetyl group. So what I'm going to do is attach coenzyme A, which is floating around in that matrix. That's one of the dissolved substances that's in there that we got from nutrients from eating, and put it in there where we're going to use it. We're going to attach that coenzyme to pyruvate and form acetyl-CoA. When we do this, we're going to break off some more hydrogens. So this is going to lose some electrons. This will be oxidized, and my NAD taxicab will be reduced by picking up some hydrogens. And these will get carried over to the electron transport chain. In this process, when I convert my pyruvates to acetyl-CoA, I always also lop off a carbon and 2 oxygens from each of these. This ends up being released as carbon dioxide, a waste product of cellular respiration. Remember, each glucose molecule was divided into 2 pyruvates, so I have 2 of these formed. So each pyruvate, 1 glucose becomes 2 pyruvates. Each pyruvate becomes an acetyl-CoA. So if we look at my inputs and my outputs, I have my hydrogen taxicab, that NAD, my 2 pyruvates that came in from glycolysis, my 2 coenzyme A's that are in the matrix from nutrients as one of the resources that I need for this process. And what comes out is my taxicab now has its hydrogen passenger, so 2 NADHs, and my pyruvate and coenzyme A come together to form acetyl-CoA. And I'm going to release some carbon dioxides. So from my outputs, my NADH gets carried, forgot to put this one, goes to the electron transport chain to drop off its passenger and then come back and continue to pick up hydrogens to take to the electron transport chain. My acetyl-CoAs go into the next step, the citric acid cycle. So the output from each previous step becomes the input to the next step. The output from glycolysis, pyruvates, come into the prep reaction. The outputs from the prep reaction, the acetyl-CoA, go into the citric acid cycle. And then my carbon dioxides are exhaled as a waste product from this. So now my acetyl-CoAs are going into the citric acid cycle. This also takes place in the mitochondria and the matrix. This is also referred to as the Krebs cycle, after the man who worked out the path of this process. Occurs in the matrix, and it begins when I take my acetyl-CoAs and I add these to a 4-carbon molecule called oxaloacetate. And when I do that, I form this 6-carbon molecule called citric acid. And that's where this cycle gets its name, citric acid cycle. You'll also see this referred to as citrate. So citrate, same thing as citric acid. Pyruvate, sometimes you'll see pyruvate referred to as pyruvic acid. So that ate and ic acid are equivalent. Citrate is citric acid. So I take my 2-carbon acetyl-CoAs, I join them with this molecule that is an intermediate in my cycle. So remember, as I go around my clock, 1 o'clock would be an intermediate, 2, 3, 4, 5, all the way to 11 o'clock. Those are all intermediates. And then I add something. I add 1 more hour. A new hour comes in, joins with 11, and gives me 12. And I'm back where I started. So oxaloacetate would be my 11 o'clock. Those acetyl-CoAs coming in would join with oxaloacetate to give me my 1 o'clock or my midnight citric acid, where I start all over again. I also have NAD and FAD. Oh, they shouldn't have their hydrogens with them in this, should they? They take, when they come in, or when they start, when they're in the process, as inputs, their NAD and FAD. They leave the process after they've picked up hydrogens and become NADH and FADH2. So my citric acid, I have 2 acetyl-CoAs coming in. So this cycle gets 1 turn for 1 acetyl-CoA, 1 turn for the second acetyl-CoA. So 2 turns from each glucose molecule that we started with, or 1 for each pyruvate, which was converted to acetyl-CoA. I have 2 acetyl-CoAs, 1 for each acetyl-CoA. I'm going to form some ATP. So my end products from the citric acid cycle, I end up with 4 carbon dioxides produced as waste product. I get 2 ATPs. I get a little bit of energy out of this. And then I've got 6 of my NAD taxicabs and 2 FAD taxicabs that will carry some hydrogens off to the electron transport chain. So let's look at this cycle a little more closely. Its name comes from citric acid being formed as the first intermediate in the cycle. That's why I get the name citric acid cycle. Citrate, or citric acid, is formed when an acetyl-CoA, coming in from the prep reaction, jumps into the cycle here with the final intermediate product, oxaloacetate. When acetyl-CoA and oxaloacetate come together, they form citrate. They drop off that coenzyme A, and they form citrate. This is my first intermediate in the citric acid cycle, and then it's a cycle. I'm going to convert it to another intermediate, convert it to another intermediate, convert it to another intermediate, to another intermediate, until I'm back to oxaloacetate, and the next acetyl-CoA comes in and forms citrate. So it's a cyclical process where each intermediate forms the precursor to the next, to the next, to the next, and I always end up with the same one, and the cycle starts over with the next input of acetyl-CoA. Here's the whole process where my other inputs come in. So my acetyl-CoA comes in, joins with oxaloacetate, forms citrate. Ooh, my taxicab picks up a hydrogen from the citrate. It also lops off a carbon and 2 oxygens that get exhaled as carbon dioxide. Some more NAD comes in and picks up some more hydrogens from this intermediate, and I get rid of some more carbon and oxygen. So exhale some more waste product, And in this process, I'm going to generate a little bit of ATP. I have another taxi cab, so here's a different taxi company, comes in and picks up a hydrogen. And this continues. My NAD, I pick up some more, and this continues in this cyclic manner until I'm back to oxaloacetate, which picks up acetyl-CoA coming in, and we continue the process again. If we look at the accounting here for the citric acid cycle, I have 2 acetyl groups. My acetyl-CoA comes in. I have 8 taxi cabs, 6 NAD taxis and 2 FAD taxis. And I've got some ADP and P that can be put together. My outputs, when I convert my acetyl groups through this process, I'm going to break off all of these carbons. I'm going to end up with 4 carbon dioxides produced. These both pick up all their passenger hydrogens. So 6 NADs, 2 FADs. This shows that FAD, it's really FADH. And I think they just do that to make it similar to remember. So it doesn't really pick up, this looks like it picks up 4 hydrogens, but it's FADH, it only picks up 1 extra hydrogen to become FADH2. And my ADPs are put back together. I get 2 ATPs out of this process. My carbon dioxides go to a waste product. They're exhaled. My taxi cabs carry their hydrogens to the electron transport chain where they will drop them off and then come back to pick up more passengers. And my ATP is ready to be used for whatever work the cell needs it for. So now we've carried all these hydrogens to the electron transport chain. Remember, when we see electron transport chain, that always takes place on a membrane. So here it's on the cristae, that's that inner folded membrane inside the mitochondria. In eukaryotes, it's on the cristae, but in aerobic prokaryotes, that's just the plasma membrane. Remember, our outer membrane on our mitochondria came from endocytosis originally, but just in regular bacteria, that's just their membrane. Most chemical reactions in bacteria take place just on their membrane through those membrane proteins. The electron transport chain, all those H's, all those hydrogens, all the electrons that were carried by our hydrogen taxi cab, bring those hydrogens with their 1 electron and 1 proton to the electron transport chain. That was their destination. And we're going to produce a lot of ATP by oxidative phosphorylation, by taking those phosphates that had been separated out from their ATP to form ADP, adenosine diphosphate, and we're going to stick them back on. And we're going to use ATP synthase again. So if you remember photosynthesis, ATP synthase, ACE, it's the enzyme that synthesizes ATP by putting the 3rd phosphate onto ADP. So we're going to see that enzyme again. We do this through a series of carrier molecules, just like we did in the electron transport chain at photosynthesis. And we're going to bounce those electrons from one carrier to another. We're going to play hot potato with the electrons. So we have lots of proteins, cytochrome, that are going to let us bounce those electrons down the line, down the membrane. At the end, this is important to remember, that oxygen that we started with in our equation, we haven't used it yet. It had to be present for us to move into the mitochondria, but here's where we're going to use that oxygen. It is our final electron acceptor. So oxygen, O, when we see electron in all these processes, we're seeing that hydrogen atom with its 1 proton and its 1 electron. If I combine H's and O's, I get water. On the product side of cell respiration, my inputs are glucose and oxygen. What comes out? Carbon dioxide and water, my waste products. So this is also, think about what we exhale. We exhale carbon dioxide, but there's also moisture There's condensation when we exhale. That's the water. So we exhale, or we remove water as a waste product. The reason that we do this, our oxygen, the reason it's present as our final electron acceptor. So if I didn't have some way to get rid of all those hydrogens that I broke off, that carried, my taxi cab carried over to the electron transport chain, all these hydrogens would build up and the inside of the cell would become very acidic. So by tacking that hydrogen onto oxygen, I form water, a harmless waste product. Right, so let's look at our electron transport chain. This looks very similar to the way it looked in photosynthesis, except in photosynthesis, I built my concentration gradient with all the hydrogens on the inside, and then they float out. Here it's going to go the opposite direction. I'm going to build up that concentration gradient outside, and then ATP synthase will let those hydrogens flow in a controlled manner to drive ATP synthase in joining a phosphate to an ADP to produce ATP that can then go out through a channel protein to be used by the cell wherever it's needed to be used by the cell. And so I come in, my NADHs come in and they drop off their hydrogens. The hydrogens get pumped out here to build a concentration gradient. So when they pass through here, they spit out the proton and they keep the electron. The electron bounces down through all these proteins here that capture a series of oxidation reduction reactions. At each step, the electron loses a little bit of energy. We're going to capture that energy to push these hydrogens out and maintain this concentration gradient. These hydrogens really want to get back here. The electrons really want to get to their hydrogens. When that electron is no longer fun to play hot potato with, the electron transport chain drops it off back in here. Those hydrogens, the protons, the hydrogen ions, my protons, flow through ATP synthase, where they get to come back together with their hydrogens, and then they bind with an oxygen to form water. So the electron transport chain, this is my main ATP producer in aerobic cellular respiration. So when we find out, we're going to follow the electrons, and those electrons come from my hydrogen. So the hydrogens that my NADH taxi cab delivered, these hydrogens, the NADH is very efficient, and it allows, they have enough energy that each one can make 3 ATPs. So remember, I had 2 NAD taxi cabs carrying 2 hydrogens from glycolysis. I had 2 NADs carrying hydrogens from the prep reaction. So there's 2 hydrogens, 4 hydrogens, and then 6 NADs carrying 6 hydrogens from the Krebs cycle. So 10 hydrogens here. Oh, that's a times 3. Each one can make 3 ATPs. I get 30 ATPs. And the FADH2s, this is FADH, and then each one picks up another hydrogen, becoming FADH2. Each one of these hydrogens only has enough for 2 ATPs, to produce 2. So I have 2 hydrogens carried by FADH. Those come in from the Krebs cycle. There's 4 ATP, 2 times 2 ATPs each, 4. So my grand total, 4 from the FAD hydrogens, 10 from the NAD hydrogens, give me 30 ATPs, 4 ATPs, give me a grand total of 34 ATPs from the electron transport chain. Then I also have 2 ATPs that I formed in glycolysis and 2 ATPs that I formed in the citric acid cycle. So I have a grand total possible of 38 total ATPs from aerobic cellular respiration. You'll see the accounting lists 36 to 38. The reality is probably closer to 30 because every now and then these taxi cabs decide that hydrogen's needed somewhere else, and they'll kind of get short-circuited or detoured around, and those hydrogens will get used somewhere else. So 38 is in the perfect world if every one of those gets shunted to the electron transport chain to where it needs to go and produces all the ATP it's possible to produce. Usually it's 36 is probably the better estimate, because normally at least 2 get shunted away, but it really could be anywhere from 30 to 38. But this is the number that you'll most commonly see, 36 to 38. And that 38 max comes from a perfectly efficient, no hydrogens are needed, no electrons are needed anywhere else, and so they all are converted to ATPs. At the end of the process, these hydrogens that were carried by those taxis and then dropped off would make the inside of the cell, of the mitochondria, and then of the cell acidic and destroy it. So we are going to neutralize it by combining with oxygen, that oxygen that came in at the beginning of the process, to form water. So our electron transport chain, just once again, the complexes, the different proteins that are embedded in that membrane, pump the hydrogen out of the matrix into the inner membrane space. So that's the space between the cristae and the outer membrane, to build a big concentration gradient. So we get an electrochemical gradient with those positive hydrogen protons on the outside wanting to get back to their electrons. We have ATP synthase, the doorway that will allow those hydrogen ions to flow down its gradient. And as they flow through ATP synthase, this is my enzyme that joins a phosphate onto ADP to make ATP. Through the process of chemiosmosis, I have a chemical gradient of hydrogens, hydrogen ions, and hydrogen electrons. It's my chemical gradient. And when those hydrogens flow through ATP synthase, they join with oxygen to make water in the inner space, in the inner matrix. So chemiosmosis. I end up with water on one side of the membrane, but it starts through a chemical gradient of hydrogens being separated between their proton and electron, and then later joining with oxygen to make water. So the ATP production is linked to this hydrogen proton gradient, or the hydrogen ion gradient. And then that ATP is able to move out of the mitochondria to be used for whatever work the cell needs it to be used for. And then we're going to recycle that. So remember our chapter 6 on that recycling of ATP. When I use it to do work, I break off that third phosphate. I get adenosine diphosphate and an inorganic phosphate, and then I come back to the mitochondria so that I can put them back together through the process of cell respiration and the electron transport chain. So again, I'm going to bring all those NADH taxicabs and FADH taxicabs in here, drop off the hydrogens. The hydrogens have 1 proton, 1 electron. When I pump those hydrogens out, I drop off the hydrogen, become NAD. My taxi's empty, can go pick up more hydrogens. I pump that hydrogen out, but I pull its electron off. The electron has lots of energy, and it's that energy, as the electron is passed from molecule to molecule down the electron transport chain, that drives these hydrogens being pumped out here and this buildup of hydrogens. When we have a concentration gradient, molecules want to flow from high concentration to low. We're only going to let them flow back here through facilitated transport through ATP synthase. And as those hydrogens flow through, ATP synthase is my enzyme that's going to connect the phosphate onto the ADP to give me ATPs that will then be pumped out into the cell or pass out into the cell to do work. Once these hydrogens are back in here, my electron transport chain lets the electron go. it can join with its hydrogen ion to become hydrogen, and with an oxygen to become water. And then my water is released as a waste product. So this is the complete 4 steps. Glycolysis out here in the cytoplasm, no oxygen involved. I put in some ATP to kickstart it. I get 2 ATPs out. My end product from glycolysis, pyruvate, goes into the prep reaction to be readied to be put into a form that can be used in the citric acid cycle. So my pyruvates are converted to acetyl-CoA, and my hydrogen taxis pick up some hydrogens to go to the electron transport chain. That acetyl-CoA comes into the citric acid cycle, which can produce 2 ATPs. It also releases some carbon dioxide, some waste product. And at the end of this, I've broken down all my carbon from hydrogen bonds, gotten rid of all my waste product, and all of my hydrogens have now ended up here on the electron transport chain, where I can generate 32 to 34 ATPs for a grand total of 34, 35, 36, 37, 38. So between 36 and 38 ATPs. So per glucose molecule, I can get 2 ATPs from glycolysis, 2 from the citric acid cycle, and 32 to 34 from the electron transport chain. This is a really, really efficient process. It might not seem like it with this efficiency of 39%. If you consider the old incandescent bulbs were about between 1% and 5% efficient, and the current ones are about 25% efficient. Your car, if you drive a really, really, really energy efficient car, might be 30 to 40%. Max, this is actually the low end. The efficiency is calculated somewhere between 39 and 45%. So it's actually very efficient because we recycle those molecules. And we do this stepwise fashion. All the rest of that energy is lost as heat. So let's do some accounting here. Our inputs to this process, glucose with 6 carbons, 12 hydrogens, and 6 oxygens, and 6 dioxides. So that gives us 18 total oxygens. Law of conservation of mass tells us what goes in, we have to account for, has to come out. Where is it coming out? So here are all our steps. Here's my glucose, my C6H12O6, and here are my 6 oxygens here going in. So those are my inputs. Let's look at those carbons. Here I have 2 carbons coming out in carbon dioxide from the prep reaction, and I have 4 carbons coming out as carbon dioxide in the citric acid cycle. All 6 of my carbons are accounted for. My 12 hydrogens, well, 2 get picked up there, 2, there's 4, another 6, 10, and 2 picked up here. 12 hydrogens are picked up by hydrogen taxicabs and delivered to the electron transport chain, where at the end of the process, they are dropped off. When they're reunited between the hydrogen ions and the electrons that were split up into 6 molecules of water, there are my 12 hydrogens at the end of the process. Check. My 18 oxygens, well, O2, 2 of those in this carbon dioxide that's exhaled. There are 4 of them. There are 8 here, 4 carbon dioxides with O2, 8. So there's 12. Well, I had 18. Oh, there's another 6 of them here that come out in my waters, for a grand total of 12 oxygens. And there we have our 6 carbon dioxides and our 6 H2Os. What happens if oxygen's not available? So remember, we didn't actually use the oxygen until we were in the mitochondria. We couldn't go into the mitochondria without oxygen though. So if oxygen's not available, then we can't move into the mitochondria. We can't have the prep reaction, the citric acid cycle, or the electron transport chain. If oxygen's not available, that pyruvate that we formed from glycolysis can't enter into the mitochondria. Our process is really efficient, and what allows us to go through this process is our recycling, our NAD. And our NAD is what's taking those hydrogens from our pyruvate into the electron transport chain, from our acetyl-CoA into the electron transport chain, and from all those intermediates in the citric acid cycle into the electron transport chain. Without oxygen, we can't go in there because our NADs can't pass in there. If they can't pass in there and drop off their hydrogens, then they're full taxis. They can't pick up any more passengers. So every time we break down more glucose in glycolysis, we have these hydrogens with nowhere to go. That would destroy the cell, so we have to have some way to deal with this. And so we have fermentation. Fermentation is an anaerobic process that happens when there's no oxygen available, and so we can't move into the mitochondria. Pyruvate is our pivotal metabolite. So in cellular respiration, when we have oxygen, our pyruvate's going into the mitochondria, it's being converted, the process is continuing, but when oxygen's not available, we can kick to this anaerobic process. We don't go into the mitochondria, we stay in the cytoplasm, but we have to have some way to deal with those hydrogens as we break down glucose to pyruvate and lop off those 2 hydrogens, and our taxi cabs aren't picking them up because they have nowhere to go. What they're going to do is they're going to go back to the pyruvate and stick the hydrogens on. So during fermentation, our glucose is incompletely metabolized. We have those 2 pyruvates, but when my NADH, when my taxi cab carries those hydrogens back, it's going to tack it onto pyruvate in different ways that will result in the production of either lactate or to alcohol and carbon dioxide, depending on what organism. So why might oxygen not be available? It might be the environment has no oxygen present. It might be because the oxygen demand is more than the cells can accommodate. So what we're familiar with is if you do a sprint workout, if you're a runner or a swimmer, you do a sprint workout, if you're working really, really, really hard and you can't catch your breath, you're working anaerobically. Your muscles are using oxygen at a faster rate than you can get the oxygen to them. They're now anaerobic. They don't have the oxygen they need, and so they can't go through cell respiration. So our muscles will kick to fermentation. Bacteria and yeast will use fermentation when no oxygen is available. So this is an anaerobic process, and we're going to reduce gain of electrons. We're going to tack those hydrogens onto pyruvate and convert it to either lactate, lactic acid. Remember, 8 is the same as an ic acid, lactate, lactic acid, or we're going to convert it to alcohol and carbon dioxide. So our NADH, our taxi, is going to transfer its electrons to the pyruvate. Alcoholic fermentation is done by yeast, and it produces carbon dioxide and ethyl alcohol. We use that to produce alcoholic beverages and bread. So when we see bread dough rising, if you're baking bread, when we put that yeast in with the flour, we're giving it its glucose, right? Flour is a starch. It's a complex carbohydrate. You can break it down into its building block parts of glucose, but it's very, very dense and the yeast will very quickly use the oxygen available to it, and we'll have to kick to fermentation. And fermentation produces alcohol and carbon dioxide. This carbon dioxide gas is what makes the bread dough rise. And when you cook bread, it smells kind of the same as if you drive by a beer brewery, because that's what's burning off as you bake your bread. The alcohol burns off, so it smells similar. Lactic acid fermentation, certain bacteria and fungus use it. Some animal cells, like our muscle cells, produce lactic acid when they work anaerobically. And we use that to produce cheese, yogurts, and sauerkraut. And then there's some other types of acid that can be acids or alcohols that are produced by different bacterial species. So don't worry about how complex this looks. This is just showing, this is fermentation really is modified glycolysis. This is glycolysis in the absence of alcohol. My glucose, I kickstart it with 2 ATPs. I split to my G3P. Same process as glycolysis, right down to pyruvate. But here, without oxygen present, typically my NADH would go off to the electron transport chain inside the mitochondria. It can't do that. So now every glucose that gets broken down, these are all filled up with hydrogens, so they're just dropping off the hydrogen. So these taxis have to do something with that hydrogen. So what they do is they grab the hydrogen off the pyruvate. They come down here, grab the hydrogen, and then they're going to kick that hydrogen. They're going to take this hydrogen and they're going to tack it onto pyruvate, but in a different place. And so in some processes, they put it on a different place that makes lactate. In some, they put it into a different place that makes alcohol. And then carbon dioxide is released. And we still get that same 2 ATP net gain, just like we do with glycolysis. So instead of dropping off the hydrogen, at the electron transport chain, from the pyruvate to the electron transport chain, they're going to drop it off. They're going to put it back onto the pyruvate. They're going to convert it to acid or alcohol. Both of these, lactic acid and alcohol, are organic molecules with lots of carbon-hydrogen bonds. So you can see we lost some carbon here. We've broken off some hydrogen, so there aren't as many carbon-hydrogen bonds as we started with. Remember, that's my potential energy. That's my chemical energy locked up in here. I've broken some of those to release a little energy. Cellular respiration breaks every carbon-hydrogen bond. There's no more potential energy left in those carbon-hydrogen bonds because there aren't any of those bonds left. Here I still have some of those carbon-hydrogen bonds. So there's potential energy left in the molecule. So this is much less efficient than cellular respiration because I'm leaving energy on the table. The advantages, though, are I can get a quick burst of ATP for muscular activity. So it gives me energy for a really short period of time, even if oxygen is lacking. So it's a stopgap measure. The disadvantages are those end products are toxic to cells. Lactic acid changes the pH, and that causes in your muscles, it causes muscle fatigue and an oxygen debt. And it can only get carried on for so long. The alcohol produced in fermentation actually kill the yeast. So there's a limit to how long I can use fermentation as an energy source if I'm an aerobic organism. Efficiency-wise, it's just not nearly as efficient. I get 2 ATPs. It's equivalent of 14.6 kilocalories. But aerobic respiration, the complete oxidation of glucose, where I have no carbon-hydrogen bonds left, gives me 686 kilocalories. So it's really only 2% as efficient because I only get 2 ATPs instead of the 36 to 38. So here, if I do my accounting, my input is glucose and some ADP plus P. My output, either 2 lactic acids or 2 alcohols and carbon dioxides, and a net gain of 2 ATPs. All right, to finish up, let's look. Our equations always show glucose as my input. Glucose is my energy source, but I can use other foods as an energy source. Right? It's not just glucose. I can use all kinds because of metabolic reactions. I have enzymes that can convert things to the right form. The reason we always show glucose is because that's what our cells are set up for. I always have the enzymes I need to do cellular respiration starting with glucose, so I get the most energy out of it. If I start with other organic molecules, I can still convert to an input at some phase of cell respiration and get the energy out. But I'm going to have to use energy to convert my starting molecules to a point where they can enter the cycle. So it's not as efficient. But I have lots of different foods other than glucose. So all my foods are sources of energy-rich molecules, some kind of organic molecule with lots of carbon-hydrogen bonds, carbs, fats, proteins. You can use all of these, and I can break them all down. When I break down organic molecules, so catabolism, I break down molecules, and exergonic reactions, when I break down molecules, I release energy. Synthetic reactions, I build molecules from smaller molecules. Anabolism tend to be endergonic. They use energy. So for some of these sources, if I don't start with glucose, I either have to break them down to get glucose molecules out first before I can shunt them into a part of the metabolic reactions for cellular respiration. Sometimes I have to break them down and build them into something else. So I lose some ATP. So the metabolic pool concept. Here is my perfect starting point. If I eat glucose, I start with glucose, I can go right through my process. Glycolysis, prep reaction, citric acid cycle, electron transport chain, and I get back my maximum amount of energy. But if I start with other carbohydrates, where they are polymers, polysaccharides of monosaccharides, that are all pretty similar to glucose at some level. So I can break these down. I can have catabolic reaction through hydrolysis, break them into the building blocks that are glucose or something similar. Will let me jump right into this process. It'll take a little longer. I'll have to break some molecules down first, have some enzyme activity in here, and then I can go right into the process. So when I eat carbohydrates, this is pretty straightforward. My fats are made of glycerol and fatty acid. So glycerol, I can actually convert and break down into pyruvate and then jump in at the glycolysis point, at some point in glycolysis, one of those 10 steps, and continue on and get energy out. Not nearly as much. I miss those ATPs here. My fatty acids, I can convert through a number of steps into the prep reaction, into acetyl-CoA at the prep reaction, and then continue with the process. So again, I'm circumventing part of the process, losing some of the energy, but I can still get energy. Proteins, I can break my proteins down into their building block amino acids, those 20 amino acids. So if I break down my big molecule to smaller ones, depending on which of the 20 amino acids I get, I can jump into a late portion of glycolysis and end up with pyruvate and continue in the process. I can jump into the prep reaction, forming a part of acetyl-CoA and continue with the process. Or I can form one of the intermediates from the citric acid cycle and jump in here. For all of these, for protein, I always have to get rid of the amine group, the nitrogen-containing group, because the amine group will build up and form ammonia, which is a toxic compound. So nitrogen's building up, become ammonia, are toxic to cells. So I have to break those off, deaminate these, and lose them through the kidneys in urination. My nitrogenous wastes are released that way. So proteins are really our kind of worst bet for getting energy. Carbs and fats are good energy sources. Proteins, we need proteins to build muscle. But excess proteins, we have no way to store these. We have ways to store carbs and fats. We have no way to store proteins. So if we need to rebuild muscle, if we need to rebuild enzymes, if we have used up lots of proteins, we need to replenish them. We have no way to store them though. So if we excess proteins, we have to process those and get rid of the ammonia compound waste products that are toxic. So these are the most energy intensive to process in order to get energy out of, and give us the least amount of energy because of that, and put a high strain on our kidneys. So for a protein source, protein, yes, when we need to build muscle mass, when we need to replenish our proteins, but not a good source of energy. So that's our metabolic pool concept, is that all of our foods can be shunted into this process for energy at some point. But other than glucose, which is what we're set up immediately to start, all the others we have to do some processing for. So if you remember the building blocks of our biomolecules, you can work out these processes here. And that is it for cellular respiration. And this takes some time. I'd recommend going through step by step. Learn one step first. Remember that step. Know what goes in, what goes out, that accounting. What do I put into each step? What comes out of each step? So really pay attention to those slides. And then what we use those outputs for. Now the output from each step becomes the input to the next step until we have broken down our glucose completely. All right, so that is it for Chapter 8 on cellular respiration. Bye for now.